Good morning, friends. Today I am going to discuss about a new topic, that is hydrolysis of amide bond or peptide bond, which is mediated by a classical enzyme termed as chymotrypsin. Chymotrypsin is a classical enzyme which belongs to serine protease category. So this particular topic is covered under bioorganic chemistry course, that too under module 2, how enzymes play a major role in organic transformations, that is enzymes in organic transformations. So today's topic is how chymotrypsin mediates the hydrolysis of a peptide or amide bond. So before entering into the topic, you can see there are a variety of enzymes which are involved in amide hydrolysis or peptide bond hydrolysis, where you can see you can classify the enzymes which are involved in hydrolysis of amide bond or a peptide bond. This amide or peptide bond is present in a protein. So enzymes which are involved in hydrolysis of proteins are actually termed as peptidases, or otherwise they are also termed as proteases. You can see the hydrolysis which occur from the amide bond, or the hydrolysis of amide bond from the C terminus side. So now here you can see this is the hydrolysis of amide or peptide bond which occurs from C terminus. For any protein there will be two terminus. The starting point will be N terminus, the ending point will be C terminus. So now you can see this is the starting point, so this could be the N terminus for this protein, and you can see the protein is lengthened, it is growing and growing. Here the free carboxyl group is not shown, but it should end over here. So this is the free amino group, it should be the first amino acid, and here there should be COO minus. So like this, now here you can see this is the free N terminus starting amino acid, this is the ending amino acid where free carboxyl group. So any protein will have N terminus and C terminus, free N terminus and free C terminus. So if hydrolysis of an amide or peptide bond occurs from the N terminus side means you can see this is the carboxyl group, this is the amino group. Now this amino group should belong to this amino acid. So carboxyl group belongs to this amino acid 1, amino group belongs to amino acid 1. Now you can see this carboxyl group should belongs to this amino acid, amino group belongs to this amino acid. So assume if this is R1 and R2, amino group and carboxyl group belongs to R1, first amino acid, amino group and carboxyl group belongs to second amino acid. So what is the meaning? If hydrolysis of a peptide or amide bond occurs from the C terminus side, right? So that means the C terminus should be contributed by that particular amino acid. So here you can see this is the C terminus, the carbonyl group contributed by this amino acid. So you can see CO and NH, this particular bond, right? So where I can see, so this particular bond, so hydrolysis of this particular bond, right? So here the carboxyl group is contributed by this amino acid. Hydrolysis of amide or peptide bond from C terminus contributed by a particular amino acid. So that can be denoted as P1 and P2 like this. Now this CO belongs to this amino acid, this CO belongs to this amino acid. So the bond to be hydrolyzed should be like this. So this bond should be hydrolyzed and this bond should be hydrolyzed. So in one way your enzymes will target the amino acids in the protein where the carbonyl group should be contributed by that particular amino acid, right? In another term, here you can see hydrolysis of amide or peptide bond contributed by the amino group. Now again it should be the same. Now this is the amino group contributed by this amino acid, right? So from amino side means again the same bond will be targeted. Here you can see the same bond will be targeted. So amino group, this is the amino group contributed by this amino acid. This amino group should be contributed by this amino acid, right? So this amino group should be contributed by this amino acid, right? And this amino group, free amino group, should be contributed by this amino acid. So like a lot we can put, should be like this. This should belongs to what amino acid? Belongs to one amino acid. Now this lot should belongs to another amino acid. So amino group is contributed by this amino acid. Amino group is contributed by this amino acid. Amino group is contributed by this amino acid. So if you target, if enzymes target like that, again the same peptide bond will be targeted. Same peptide bond will be targeted. So the concept is enzymes can either target an amino acid whose carbonyl group should be donated, or enzymes can target amino acids whose amino group should be targeted by an amino acid. So that's it is termed over here. The target could come from C terminus side, or the target of your enzyme can come from the N terminus side. So don't worry about all these things. These are all basics. You can classify the proteases or peptidases into majorly two types. One is exopeptidase or exoprotease. Another one is endopeptidase or endoprotease. Now here you can see some of the sorry, target amino acids by the endopeptidases. So we will take endopeptidase for today's understanding. These are the target amino acids. You can see these are all set of the enzymes, endopeptidases or endoproteases. You can see trypsin, protease from submaxillary gland of your mouse. You know, submaxillary gland is situated in the salivary gland. Chymotrypsin, staphylococcus, V8 protease, and aspartate and protease, pepsin, endoproteinase, and you leave the cyanogen bromide. It is a chemical. Now today we are going to discuss about a chymotrypsin. What are the target amino acids in the protein? That means enzymes are very specific. They will not target any amino acid in a protein. So what is the specificity of chymotrypsin? Chymotrypsin will target a protein, and you know proteins are made up of 20 amino acids. So any amino acid will not be targeted by your chymotrypsin. Very specifically it will target three different amino acids, that is phenylalanine, tryptophan and tyrosine. And remember, the peptide bond generated by your phenylalanine or tryptophan or tyrosine in a protein, right, that should be contributed from your from the C terminus side. That is, the carbonyl group of the peptide bond should be donated by, or it should come from phenylalanine or from tryptophan or from tyrosine. So here C means the carbonyl group in a protein should be contributed by either phenylalanine or tryptophan or tyrosine. If it was contributed by any one of these amino acids, that particular CONH peptide bond will be specifically targeted by your chymotrypsin. So that is the meaning of the C. If it is N, the amino group CONH, NH, the amino group should be contributed by either aspartate or your glutamate. Now here you can see pepsin, an enzyme which is secreted by the gastric gland, where phenylalanine, tryptophan, tyrosine, the same three amino acids which is targeted by your chymotrypsin, right? You see, whereas there is a slight difference. Your pepsin will target all the three amino acids, but not from C terminus side, from N terminus side. That means the amino group should be contributed by either phenylalanine or tryptophan or tyrosine. So please understand, any enzyme, they are highly specific in nature. So proteases means they cannot just like they target any protein or protein with or any amino acids. So chymotrypsin can target any protein, but just like that it will not hydrolyze all the peptide bonds. Only peptide bonds contributed by these three amino acids only or specifically or selectively targeted by chymotrypsin. So specificity is a unique feature of enzyme. So you can see some list of enzymes which play or their cleavage points or how they interact with protein. Next, you see, before entering to understand the role of chymotrypsin, three significant peptidases I have listed. One is pepsin, another one is trypsin, third one is chymotrypsin. There is also carboxypeptidase A and B, which I'm not going to discuss. Here you can see pepsin is an important enzyme which is secreted by the gastric portion of your stomach. Exclusively chief cells of your stomach will secrete pepsin in the form of pepsinogen, that is the zymogen or inactive part. This pepsinogen will be converted into pepsin, and now pepsin will act upon any proteins what you are ingesting. Your food contains many protein, and those proteins in the stomach will be effectively hydrolyzed into small pieces, small oligopeptides and peptides with the help of your pepsin. And the speciality of your pepsin is your pepsin always work well in an acidic pH. You know, stomach pH is 1.2 to 2.2 or 1.4 to 2.4. That is approximate pH range. Similarly, another two important enzymes, that is your trypsin and chymotrypsin. These two enzymes were actually secreted by the exocrine part of your pancreas. These two are pancreatic enzymes, and these two enzymes were synthesized in an inactive format termed as trypsinogen and chymotrypsinogen. When food enters into small intestine, chymotrypsinogen will be converted to chymotrypsin and trypsinogen will be converted to trypsin. Now active forms of your trypsin and your chymotrypsin. Now these two are the active forms. Now both will act upon the proteins present in the food, and those proteins will be hydrolyzed into small and short oligopeptides and peptides. Now there is a big and drastic difference in the pH, right? Now these two enzymes are highly active in mild alkaline pH, whereas pepsin is active under high acidic pH. When you transfer pepsin into alkaline pH, it will denature, pepsin will not work. Similarly, when you transfer trypsin and chymotrypsin into an acidic pH, they will not work. So even though you can see the the target of your pepsin is similar to the target of your chymotrypsin, but still there is a change in the pH. So even though the target amino acids are same, pH is totally different, right? So if you want to understand the mechanism of action of chymotrypsin, first of all you should understand the role or physiology of chymotrypsin. So I have given 3 YouTube links. I request you all to just watch the videos, right? A very simple digestion process is explained so that you can understand the significance of these 3 enzymes. Now we will enter into the real topic, chymotrypsin. What is the role of chymotrypsin? It involves in hydrolytic cleavage of peptide bonds or amide bonds. Chymotrypsin is a classical example for covalent as well as acid-base, general acid-base catalysis. General acid-base means either HCl or H2SO4 or any other simple organic molecule can act as an acid or base catalyst. So that is general. If very specifically we are using water, that will become specific acid-base catalysis, or otherwise any catalysis could be general. So chymotrypsin is one such enzyme which follows 2 different mechanisms, right? It really explains how a general acid-base catalysis occurs. It's a classical example. Similarly, when you see the biochemical reaction mediated by chymotrypsin, you can see the formation of a covalent bond. In that way, chymotrypsin is a classical example to explain the modality or principle of covalent catalysis. So 2 different concepts occurs. Acid-base catalysis occurs as well as covalent catalysis occurs. And already we have discussed, right? Your chymotrypsin can act upon any protein. Please remember, it can act upon any protein, but your protein may be made up of different amino acids. Already we have studied 20 different amino acids are there. So your chymotrypsin here, the specificity restricts with the amino acids, not with the protein. It can act upon any protein, but It cannot act upon any amino acids present in the protein. Very specifically, peptide bonds contributed by phenylalanine, tyrosine, and tryptophan. Right? Very specifically, very specifically, these three, right? Peptide bonds, peptide bonds contributed by phenylalanine, tyrosine, and tryptophan will be targeted by your chymotrypsin. That is the specificity. So remember, specificity of chymotrypsin is not with the protein. It can act upon any protein, right? Then where comes the specificity? Those substrate proteins, those target proteins should contain at least any one of these amino acids, right? Suppose if there is a protein like this, right? And assume the protein is made up of, say for example, alanine. The protein is made up of, say for example, glycine, cysteine, and serine. Now here there is a peptide bond. There is a now here there is a peptide bond. There is a peptide bond. There is a peptide bond. Now the question whether chymotrypsin will act? No. Why? Chymotrypsin will approach this protein. It will look for the presence of these three amino acids. There is no phenylalanine, there is no tyrosine, there is no tryptophan. So automatically, what happens? Your chymotrypsin cannot hydrolyze this protein, right? Whereas there is another protein, right? There is another protein like this. Say assume there is another protein. So this protein, right? Say contains, say it contains say glycine, it contains say tyrosine, it contains say lysine, it contains alanine. Now the question whether chymotrypsin will act, right? Glycine, we will see, right? It will act. I will extend this, right? So here again there is alanine. Now the question whether chymotrypsin will act, yes, because there is the target amino acid Y. So where the hydrolysis will occur, so the carbonyl group will be, this is the amino group. So this is the amino group and this is the carboxyl group, right? So now what happens? The amino group was contributed here. The carboxyl group was contributed by tyrosine over here. Now hydrolysis occur over here. So now how many simple peptide you will get? You will get two peptides. One with A, G, Y, another with K and A, right? So two things you have to remember. Chymotrypsin, it will go and it will try, but there is no the target amino acids. Either phenylalanine, tyrosine, tryptophan is absent, so it cannot hydrolyze. Now this is another peptide. Chymotrypsin will approach this. It will see, yes, target amino acid tyrosine is here, and the carbonyl group is donated by tyrosine over here. Now this bond will be hydrolyzed. So very specific chymotrypsin, right? It looks for three different amino acids. Now the fourth point here you can see, even though we are telling hydrolysis, water will not directly involve, indirectly it will involve. So in the biochemical mechanism we will see. Now the entire reaction mediated by your chymotrypsin is divided into two phases. One is acylation phase, another one is deacylation phase. So what is acylation phase? Formation of a peptide carbonyl carbon with ester linkage to the enzyme. That is acylation. 50% of the reaction will get over. The rest you have to hydrolyze the ester linkage, you have to regenerate the enzyme. So that is deacylation. So two steps remember in chymotrypsin mediated biochemical reaction. One is acylation, another one is deacylation. So here for your understanding, I took the three-dimensional picture or three-dimensional image from protein data bank. It is an open source. Anybody can go, right? Just you type Google it, you type PDB or protein data bank. You can get any structures if it is deposited. Now here the blue color line indicate only the backbone. There is no side chain. right? So there is no side chains, only the backbone. You can see three important amino acids are essential for chymotrypsin activity. Chymotrypsin is an enzyme. It involves in biocatalysis. It will act as a catalyst. So when you say an enzyme as a catalyst, right, all the amino acids in an enzyme will not involve in catalysis. Only very few amino acids will involve. Three such amino acids were identified in chymotrypsin. So you have to remember these three amino acids with position wise. It's very important. Serine 195, histidine 57, aspartate 102. So here you can see this is serine. This part is serine. So the red color ball indicates oxygen, right? The blue color ball indicates nitrogen of histidine. So serine 195, histidine 57 and aspartate 102. See, position is different. 195th position serine is present. 57th position histidine is present. 102nd position aspartate. So due to folding of protein, your proteins will be folded. They are not linear. So during folding, right, you see 57th position, 102nd position and 195th position, they come very close to each other. If you assume the structure in a linear form, 57 will be somewhere else, 195 will be somewhere, 102 will be somewhere, so they will be distant. So understand, this is actually termed as protein folding. You can see it is not linear, right? The protein is not linear like this, straight. It is not like this. So it is folded into some conformation, right? So for this folding makes distant amino acids to come very close. So in that way, 195th position, 57th position, 102nd position amino acids were brought very close. So this is a picture of chymotrypsin from one mammalian species. Here I have projected these three amino acids with only the backbone, and these three amino acids in chymotrypsin is actually termed as catalytic triad. Catalytic triad means three amino acids which play a major role in the hydrolysis of peptide bond. So you have to give much more importance to these three bonds. Now I have projected in the same chymotrypsin in a very different way. In your biochemistry course, you will be studying about alpha helix and beta pleated. So I'm not going to touch. This is a folded conformation of your chymotrypsin projected in another view, right? So here again you can see histidine. You can see this ring form, imidazole ring. Histidine 57, serine 195, aspartate 102. All these three are very close to each other. So here you can see the protein is folded, and again you can see it is folded into this conformation, and it is moved. So like this, folding. So again, same thing I have projected over here. You can see serine 195, aspartate 102, histidine 57. Assume that this is actually an inhibitor, but assume that this is the structure of protein. This yellow color, assume this is the structure of protein, and assume this could be the peptide bond. So where your serine 195 will catch hold this peptide bond and it will degrade, right? Now we will go to the mechanism of action of chymotrypsin over here. So you can see chymotrypsin, the first phase is acylation. You can see serine 195, histidine 57, aspartate 102. Three amino acids were present in chymotrypsin. These three amino acids play a major role in the catalysis of the protein, or these three amino acids are responsible to hydrolyze the protein. Now this is the substrate, this is the protein moiety, right? So first here you see there is no direct role of aspartate 102. Aspartate 102 is responsible to form a salt bridge or ionic interaction with this histidine. It will stabilize histidine, right? It will stabilize histidine. And you see histidine again in terms, it forms hydrogen bond with serine 195. So these three amino acids were stabilized. They are giving helping hand to each others in order to fix in a proper orientation. So you cannot see aspartate to be directly involving in the hydrolysis of protein. So the role of aspartate 1 or 2 is just to stabilize histidine 57, that's all. So it is only your histidine 57 and serine 195, that too serine 195 exclusively play a major role in the hydrolysis of a protein, right? So now this is the polypeptide substrate, right? Now this is going to fit into this pocket of your enzyme. So this blue color circle denotes chymotrypsin free enzyme. This is the catalytic site of chymotrypsin. The catalytic site is occupied by three amino acids: serine 195, histidine 57, and aspartate 1 or 2. Not only that, there is also another additional glycine, I hope 193 or 194, that is also involving, right? Not directly, indirectly this glycine involves. Now in this hydrophobic packet, this particular protein or peptide or polypeptide will fitted, right? When it fit, what happens? You see, initially what happens? Now this is substrate, this is enzyme. Now you got enzyme substrate complex. Now what is the role? Very carefully here you see, right? Serine contains side chain as OH. Already we have studied. In this OH group, this oxygen will act as a nucleophile. So if some atom should act as a nucleophile, means some atom should act as an electrophile. So in this OH, initially what happens? Oxygen of this serine will grab the electron from the hydrogen, and the hydrogen of serine will be released as proton. So serine will be having like this. So this is, I will make, try this short one. It's not good. So this is your serine. So serine is having what? CH2 and OH. This is the side chain, right? So this serine is located in which position? 195, right? Where here you can see this part. Now what happens? There is a bond, right? So this electron. So this electron belongs to oxygen, and this electron belongs to hydrogen. So what happens, since oxygen is more electronegative, this oxygen will grab this electron which belongs to your hydrogen. So two yellow color dots you can see. One yellow color dot is an electron, belongs to oxygen. Another yellow color dot belongs to hydrogen. Since oxygen is more electronegative than hydrogen, oxygen, what it will do? It will withdraw, or it will attract the electron that belongs to hydrogen, right? So as a result, what happens? Very simple. This hydrogen will be disassociated as what you can say as proton, because it loses its electron. You can see over here. So now oxygen is, now here you can see oxygen. Now this oxygen is actually electronegative, right? And this oxygen is actually nucleophile. It is having two electrons, two yellow dots. It is having two electrons. So that is what happening over here, right? So initially your oxygen, nucleophilic oxygen will be created in the serine, and the hydrogen will be disassociated as a proton. Now this will act as a electrophile. Now the released electrophile over here, this is attracted by histidine, right? Now you will be having histidine over here. This is your histidine. Now what histidine? Histidine is having N, nitrogen. So right now nitrogen will be having lone pair, right? Now the disassociated proton from serine, now this will act as an electrophile. Nitrogen in the histidine 57 will act as a nucleophile. So what happens? A bond will be formed between nitrogen of histidine and the dissociated proton. Hope you understood this. So friends, you need a lot of patience to understand the biochemical reaction. Before that, you should have a very good basic background about organic chemistry, exclusively what is a nucleophile and what is an electrophile. Before telling a nucleophilic attack, when we tell as a nucleophilic attack, there should be an electrophile. So a system will first generate an electrophile. When you generate an electrophile, automatically a nucleophile will be generated. Now this nucleophile can attack any electrophiles. So now the electrophile hydrogen generated by your serine was grabbed by the nucleophilic nitrogen of your histidine, or in other words, nitrogen of your histidine make a nucleophilic attack over here. Now here you can see NH, right? So I will erase. I hope you understand. So will clear. Right? So now what happens? Now you got the nucleophilic oxygen of your serine. This will make an attack to the peptide bond in the protein substrate. So you see oxygen is actually nucleophile. That means in the peptide bond you have to create an electrophile. You have to create an electrophile. So the carbonyl oxygen, the peptide bond is made up of what? CONH, right? So your peptide bond is made up of CO and NH, right? So now here there is serine O. Now the serine oxygen is having what? It is having lone pair of electron. Two electrons are present. So now this is the protein substrate. So now what happens? This has to make an attack over here. This has to make an attack over here, this carbon. So before that, what? This is a nucleophile. So you have to create an electrophile. So how an electrophile will be created? You see, when you compare oxygen and carbon of a peptide bond of this carbonyl oxygen, oxygen is more electronegative than carbon. So what oxygen will do? It will grab or steal electron that belongs to carbon. So like this. So the electron will move over here. Electron will move over here. So what could be the form? Now the form will be like this, right? So the electron has moved. So as a result, now this oxygen will have a lone pair of electron. Now this will act as a nucleophile. Similar, both are same. Now what happened to this? It has lost. This carbon has lost an electron. So automatically it will become a carbocation. Now this is a plus charge. This is a carbocation. So now this is an electrophile. Now already serine nucleophile is generated. Now serine 195 nucleophile, the oxygen, which is a nucleophile, will make an attack. It will make a nucleophilic attack to the peptide bond, exclusively carbon present in the peptide bond of your protein substrate, right? And then you can see the entire thing is stabilized by your glycine, and again same serine 195. Serine 195 side chain and serine 195 amide group is responsible for formation of the or stabilization of the entire thing. You can see tetrahedral intermediates. So this is actually termed as oxyanion hole, right? You can see this is one bond, number two over here, right? So this is this bond 1, this is bond 2, bond 3 and bond 4. So your carbon is surrounded by 4 bonds. What we are telling is an tetrahedral intermediate. And again the reverse reaction will take place where this small part of the protein, right, will be fragmented. That is, the peptide bond is hydrolyzed and you got the product. So we are telling hydrolysis. So far we didn't discuss about the role of water, right? So in the first phase is acylation. Now here you can see this part, again this piece of the protein still resides in the enzyme. So you consider this as an acyl. So that's what we are telling acylation. So now once a protein is fragmented, splitted into assume two parts. One part was released, another remaining 50% of your protein still resides in the enzyme. So with this, the acylation part is over. So what the next phase is deacylation. That means we have to remove the another 50% of your protein and you have to regenerate the enzyme as such. Right now we have changed the enzyme. The enzyme conformation is changed. Right. So now what is the phase 2 deacylation, right? Again here you can see this is the 50% of your protein. The first 50% was removed, the second 50% is present, right? Reverse reaction will take place. Again reverse reaction, again nucleophile and electrophile. Now here instead of serine, water will act as a nucleophile. In the first phase, serine act as a nucleophile. In the second phase, water will act as a nucleophile. Again here you can see OH, the oxygen present in water act as a nucleophile. Hydrogen will be released as a proton, that is electrophile. Again history repeats, same thing. Finally what you can see, the enzyme is regenerated and the remaining 50% of your protein fragment will be also released as product too. So two things you have to remember. The first phase, serine act as a nucleophile where a peptide bond is hydrolyzed. In the second phase, water act as a nucleophile where the rest 50% of the protein is also released. And we are forming a tetrahedral. In that way you can tell this is an acid base catalysis, and there is a formation of tetrahedral structure, which is a classical example for covalent linkage, right? So here the complete process of the please watch this particular video where you will get a better idea how chymotrypsin, this is a simple animation video, so you will get an idea how serine is acting as a nucleophile, how water is acting as a nucleophile. So what is the synopsis? Chymotrypsin is a classical enzyme. It is a good candidate to understand the acid base catalysis, that is general acid base catalysis, as well as covalent catalysis. It is an enzyme which involves in the degradation or hydrolysis of proteins exclusively in small intestine. So the same mechanism, what we have investigated, what we have studied so far, or what we have discussed so far, the same mechanism is also adopted for any serine proteases. Trypsin follows the same thing. Pepsin also follows the same thing. If you tell or if you say any candidate as serine proteases, any serine protease will follow the same mechanism, right? So it could be a little bit difficult or it could be a little bit technically overloaded. Don't worry. If you have any queries, if you have any doubt, you can clarify in the doubt clarifying session. So before understanding this particular stuff, please refresh what is a nucleophile, what is an electrophile, what is a general acid base catalysis, specific acid base catalysis. With this background, you approach this pretty well, you can understand. Thank you. Thanks a lot.